big molecules don't walk in. Welcome to section 2.4 TEL, AP Biology, Membrane Permeability.
Let's talk about the reason why glucose was stopped at the door of the cell bar, essentially. So when we have cells, we have a closure that separates the inside of the cell with all the organelles from the outside of the cell, which basically can be other cells or an external environment that contains other substances. So remember, inside the cell, we have your mitochondria, we have your nucleus. We basically have these organelles that work as a factory, and we need to ensure the safety of what's inside that cell. And to do so, we have something called the plasma membrane, which basically is creating the circular-like barrier that protects the inside from the outside. That factory basically needs to have its own internal balance, and that's called homeostasis. So, homeostasis is the meaning that we have this balance from the inside and outside of the cell. So essentially, when we have the extracellular environment, we're talking about that outside space, and the intracellular environment, which is our cytoplasm, which contains all those organelles inside. The goal is to make sure that we have some substances cross easily, while others require a set of proteins. So sometimes you'll see them embedded with these proteins that we're going to talk about in a second. But the goal is the environment that the plasma membrane makes basically essentially is a barrier from the cell protecting the outside environment from the inside environment and the inside environment from the outside environment. So the goal is to protect what we want to come inside the cell and to ensure that nothing is leaving outside the cell unless it needs to. And the only things that are coming inside are only good things on behalf of the cell. To do so, we have the phospholipid bilayer. And basically, the phospholipid bilayer has these phospholipids in it. And it creates a sandwich-like shape that we talked about previously. And the sandwich-like shape is important because what happens is on the outside of this bilayer, basically our bread portion, which basically is this part right here with the circles and this part right here, which both face the inside, which is the cytoplasm, and outside of the cell. So essentially what happens is we have our cell, and there's water here, and there's water here, or fluid-like structure, which means that these white circles on the phospholipid that create the phospholipid bilayer. So we have the phospholipid bilayer and we have the phospholipid head, basically have to be hydrophilic. And hydrophilic, think Philadelphia. So water loving has to be on both sides. So this side is water loving and this side is water loving. Now, inside the phospholipid bilayer are the tails, which are considered hydrophobic, water fearing. And I want you to think about that for a second. Say you have a water-like substance that needs to get from one side of the cell to outside of the cell, so from inside to outside. If it's going to go through this barrier, it's not going to want to stay inside that little spot. It's not wanting to stay in that hydrophobic area. It's going to want to go to the other side. So the reason why the phospholipid bilayer is formulated in such a shape is to protect things from really going outside of the area. Basically going, okay, I'm going inside the cell and I'm going to the other side, and then essentially saying, all right, well, I'm going to ensure that it's not going to stay in my hydrophobic environment. So that's why the sandwich-like shape of the plasma barrier or plasma membrane exists. Let's pause for a second. The fluid mosaic model is not as perfect as you think. We talked about the phospholipid bilayer and how there's a hydrophobic inside and hydrophilic outside within our sandwich. So if you remember, we have phospholipids and they form this bilayer, and on either side they're hydrophilic, and inside they're hydrophobic. That makes different types of items pass along the cell, either internally or externally, to maintain homeostasis. However, if you notice, the fluid mosaic model also has other things like proteins or glycolipids and glycoproteins. These all work together to ensure that various different types of things, I say things, that need to enter or exit the cell can do so with the assistance of these items. It's not a one size fits all in terms of traveling within or leaving the cell. So keep that in mind as we talk about the next part in just a second.
Aquaporin Express, here I come. VIP only, baby. Hang on, I'll get through eventually. Right path, right speed.
Let's talk about diffusion across the plasma membrane. In that little video clip there, you saw that there are various items that were trying to get across the plasma membrane, but they had to tell the bouncer what type of product they were. For example, if you have a small nonpolar molecule, it doesn't really need assistance from something like a protein. Instead, it's going to be able to really transport across the very basic plasma membrane that has the phospholipids that form that little sandwich. So you don't need the proteins and you don't need things like glycolipids or glycoproteins to help assist across that membrane. So that occurs for really small nonpolar molecules. You want to think of nitrogen or oxygen or even carbon dioxide because you have to think about gases and how easily gases travel through. These are all example of gases, and they're not going to take that much effort to travel across a barrier. Think about if you have a box and you wanted to open that box. That box is going to have different types of air inside, and that's because air can travel very easily. However, it's going to be a slow transport. Gases in general, such as nitrogen and oxygen and CO2, while they are efficient, meaning they don't need any sort of protein to cross, they are going to be slower to transport. Now let's talk about polar charged molecules. Those are going to be faster transport, and they do need assistance of protein, something like a channel protein to help assist the product from going from one side to the next. It's very quick, but not as efficient because it does require something like a channel protein to go through. Let's talk about water. Now, remember, we said small water molecules can travel with ease. However, if you need to have large quantities of water travel across that barrier, you need something called an aquaporin. And an aquaporin allows for faster transport. So remember, small nonpolar molecules move freely through the membrane, while large polar charged molecules or charged molecules require some sort of transport protein to move across the membrane. So the larger the product, the more likely that it's going to need some sort of assistance, but it's going to be faster. So that's the relationship between the two. It requires some help, but fast, don't require any help, slow. And you can think of diffusion as a way that works based on the size or the composition of that molecule itself.
I make you look fluid.
Yeah. Yeah, you're rigid. Yeah, you're right. I'm rigid.
Let's talk about selective permeability. With the phospholipid bilayer, remember we have a phospholipid that allows for different types of items or molecules to pass through depending on what it is. Cohesively, the phospholipid bilayer allows these small nonpolar molecules such as oxygen or carbon dioxide to easily pass through. However, when we have large molecules such as glucose or ions, they're not going to pass through with ease. What we need instead are these transport proteins, and they look something like this. Now you could have different types of channels and different types of carrier proteins. Anyway, these polar or large molecules, such as large amounts of water, which utilize aquaporins or ions or glucose, pass through utilizing these channels or carriers. So that's the second way the phospholipid bilayer is unique to allow for different structures to pass through. Remember, the phospholipid bilayer needs to be flexible. To maintain the flexibility, we need something known as cholesterol. And so cholesterol allows for the phospholipid bilayer basically not to be rigid and creates this shift motion in between the phospholipid bilayer to allow for flexibility and efficient transport of those molecules. Lastly, we have carbs. If you notice, we talked about glycolipids and glycoproteins, all starting with the root word glyco. In those, you'll see protruding structures coming out on the outside of the cell. And that's because if you take a look at a cell, we need to be able to receive signals from the outside. And that's where glycolipids and glycoproteins come in. Glycoproteins are going to be carbs attached to proteins, and glycolipids will essentially be the glycoprotein not together, but instead glycogen or glycol or carbs essentially attached to the lipids itself. So to recap, carbs are really important. They protrude to the outside of the cell, and they can be in the form of glycolipids, which are attached directly to the lipids, and glycoproteins like we see in this image here. They're important for signaling and recognition, which you're going to fall into understanding in a later unit. But for now, just note they're not essentially for transport. They're just sort of like antennas that basically act as a signal for other cells or other things to attach to the cell to come from the outside in.
Hey, Water! My head's obsessed with you, but my tail literally can't stand you. Guess I'm just complicated.
In Unit 1, we talked about phospholipids and the understanding that phospholipids are part of the lipid group. With phospholipids, they're unique and characteristic because they have a polar head and a nonpolar tail, which eventually formulates this bilayer. In this bilayer, we note that the polar heads face outward and the nonpolar tails face inward, which means it's very unique in characteristics when it comes to that phospholipid. This is what allows for those small, uncharged molecules to pass through, while other molecules that are larger need some sort of assistance like proteins. So it's important to once again note that the hydrophobic tail, which is the characteristic inside the phospholipid bilayer, is very important because the thing is, if you have a molecule that is on one side and it needs to pass over to the other side of the cell, understand the way that it goes through is it gets to this spot right here inside that has that hydrophobic characteristic, and it basically says, hey, you know what? I don't want to remain in this spot right here, so I have to keep going to the other side. And this is what allows that hydrophobic tail to be very important. It allows those small uncharged molecules to really pass through with ease. Now, if we talk about the other barriers that exist, we have something known as the cell wall. The cell wall, please note, is crucial for cell structure and function, specifically in plants. Remember, animal cells don't have cell walls, but things like plants and protists and fungi and bacteria, these have cell walls. Now, more specifically, if we talk about cell walls, we have different types of cell walls for different organisms. So plants have something called cellulose, and that is contained in the cell walls. Archaea, which are different for different types of polysaccharides or proteins, meaning they're composed of different polysaccharides and proteins. They're contained to have some cell wall there. Fungi has something called chitin or chitin that is a cell wall, and bacteria have what are known as peptoglycan. So peptoglycan are contained in those cell walls. So basically what you need to know is their cell wall characteristics for plants, archaea, fungi, and bacteria. And cell walls provide an extra shield, especially when it comes to protection against physical damage or pathogens. That's important to note because unlike animal cells, which need that fluidity, protection for things like plants are really important that the cell wall provides because they're so dependent on that water coming through that plant that they need that extra barrier. Which leads us to the idea that they have structural support. Remember, for plants, they allow for the plants to remain their shape and provide that rigidity that's necessary and prevent that collapse, especially with water. Sometimes it prevents osmotic lysis or osmotic cell death. The thing is, organelles like plants or cells like plants have to stop water from passing through so easily. So it stops the bursting of that cell, which we're going to talk about in a later section when it flows in. They have a permeability barrier, which is really important, especially for items like water. They're an extra background that exists along the cell membrane as well. They have a filtering role, which is important for large things that occur inside bacteria. You want to be able to provide that extra layer of protection and turgor pressure. This is important, especially for plant cells. Once again, we're going to talk about turgor and osmotic words later in the section in Unit 2. But for now, that's what you're going to need to know is that cell walls do exist and they give the strength, the shape, and protection that's necessary not only for plants, but for different organelles that we talked about here. So in bacteria, they also maintain other functions, bacteria, archaea, fungi, and plants, which are listed here that we talked about as well. So that's what's important with that. Now let's try to tackle the following question that you see on the screen here. Take a moment and pause the screen, and then we'll recap the question together. Okay, so in this question, it says, which of the following best explains why water molecules, despite being polar, require aquaporins for transport across the large quantities, across the membrane in large quantities? Take a moment, pause your video, and then once completed, unpause, and we'll go over the answer together. Okay, did you select the correct answer? The correct answer would be D. So the thing is with aquaporins, you're talking about water. And the thing is with small water molecules, remember, they can easily diffuse through that barrier. Remember, the inside of the phospholipid bilayer is basically they're hydrophobic, I should say. But outside that barrier, it's hydrophilic, which is why the fluid can remain inside the cell because it touches essentially the outside of the phospholipid bilayer. To give more details, let's imagine that we have the phospholipid bilayer and we have the inside there, and then we have the other part. This is a really bad picture, and we have the tails there. This portion right here is hydrophobic, so it allows for those small molecules to pass through even with water, which is hydrophilic because it doesn't want to remain inside that phospholipid bilayer. But when we have large quantities, we require things known as aquaporins, and this allows for faster movement necessary, especially for water. So it's important to note that when you hear the word water, you think of aqua. Well, aqua means water, and this will help you better select an answer for water.
Appreciate you riding along for our cosmic journey. If today's Mayshade brought you the knowledge that slaps, make sure to hit that like button to boost that signal. Subscribe if you're locked in for our next launch. And don't miss the link tree. It's got all the goods for AP Bio, PSAT Math, and FSAT Math. Before I sign out, make sure you're all in one orbit and diving in. Killing out. Till next time.